We will hear first about a very important feature of microdosing, which is the development of tolerance about the substance or against the substance. And the other presentation will be about a placebo-controlled randomized study of very low doses of LSD. So let me first introduce our first presenter, which is Tobias Buchborn, a PhD right now, I think.
Mhm.
Yeah. And he did study psychology in Magdeburg in Germany, and I learned to know him very early on. And it was obvious that he was a very serious researcher, and he did some basic research using LSD for treating depression in an animal model. That was his diploma thesis. This was an empirical study in animals, in rats, in fact. And that was he was successful with completing this and also publishing his results. And then afterwards, he studied the repeated use of psychedelic drugs and what happens in the organism in respect to tolerance or adaptation processes to the psychedelic substance if it is more often given than one time. And so let me introduce his lecture, which will be about the pharmacokinetics and pharmacodynamics of low-dose LSD and its implications in respect to the development of tolerance.
Thank you.
Please, Tobias.
Welcome everybody to ICPR 2020. This year is an online version. My name is Tobias Buchborn. I'm a biopsychologist and I'm currently with Imperial College London. And today we would like to talk about why is it actually that when you take a drug repeatedly, that the drug would change its face over time. If you take a drug for the first times, you might have, for example, your euphoria. And if you go on to take the drug on and on, this euphoria wears off, so you become tolerant. Other effects of the drug, on the other hand, might sensitize, so they become more and more pronounced. For example, the craving for the drug. Sometimes it happens at some time that new effects will come into the arena which had not been there in the first place. And if you decide to stop taking the drug, you sometimes end up in a withdrawal syndrome, which very often in its symptoms is the sheer opposite of what you experience in the acute state. Of course, not all drugs are the same, and LSD, for example, would not check all of these boxes here, but what is it actually we do know about the repeated application of LSD? We do know that it's actually quite uncommon to take psychedelic doses of LSD at high frequency. As to this, a withdrawal syndrome is also unheard of. Something that is very prominent with LSD is tolerance. Probably a reason why people don't do it that often. If you take psychedelic doses of LSD 4 days in a row, you are next to completely tolerant by the fourth day. And as quickly as it comes, it also seems to go. 3 to 6 days off of LSD seems to be sufficient to reignite the original intensity of LSD experience. Back in the days, they applied LSD even for 84 days. And by the end of this time, they exchanged LSD to a placebo, and the subject would not even recognize that it was exchanged. That rigorous tolerance seems to be. You should keep in mind, though, that these human studies were done back in the days, and they don't really measure up with modern standards. So there's still a lot we don't understand, we don't know about tolerance to LSD, and that's why we wanted to add a little bit to the understanding. Particularly, we were interested in the mechanism of tolerance. A very important mechanism is the pharmacodynamic one. And it refers to the fact that the target receptor of a drug, after some time, would stop responding to the drug. This is called desensitization, and there are different phases factors that contribute to such a desensitization. The receptor might be uncoupled from its signal cascade. It might be removed from the membrane, internalized, and it might be degraded, and the synthesis of the receptor might be inhibited. We know that LSD psychedelics act primarily by activation of serotonin 2A receptor, and we also know from animal studies that this 2A receptor can form complexes with the mGlu2 receptors. So if we're interested in tolerance, then it makes sense that we look at these receptors and adaptation of these. This is what we actually did. You see the overall project. We will not be able to speak about all of this today, just to give you an idea how complex this phenomenon indeed is. When investigating tolerance in animals, of course, you cannot really ask the animals for their conscious experience. That's why you need to look at behavioral output and somatic markers. We here decided for 2 animal models, the shaking behavior. Let me show you how it looks. That's how it looks. And for hypothermia, increase in body temperature. We chose these models because they are primarily related to the serotonin 2A receptor, as is psychedelia, and we can find both in humans as well. We started off with the dose response curve. We wanted to use the smallest dose possible, but of course still see something. And we learned that when we use this small dose and we put the animals together with their familiar littermates, they would respond better. That's why we continued that way. We used this dose, reapplied it at 4-hour, 8-hour, and 24 hours interval, and learned that the longer the interval is of the reapplication, the less likely animals become tolerant. We then went on to apply LSD on several days in a row. If you apply LSD once or twice per day, animals would not become tolerant. Only if you increase the dose at 3 occasions during this regimen to medium level, they would become tolerant. So there's a dose dependency here. We also have similar results for DOB, which is another serotonergic hallucinogen. We were then interested in whether different effects of LSD would show different tolerance development. We learned that LSD would increase the body temperature of rats when the environmental temperature is higher than the usual room temperature. And this effect can be blocked by ketanserin, so it's a serotonin 2A specific phenomenon. We then applied LSD in the same way. We found tolerance for the psychedelic signature move, for the shaking behavior, but we learned that animals would not become tolerant to hypothermia. Only if we increased 2 additional dosings during this regimen, they would become tolerant. So there is again a dose dependency here and obviously a biased tolerance development. We then had to look into the frontal cortex because it's very important for the psychedelic effect, but also believed to be important for the psychedelic signature move. We had to look at the serotonin 2A receptor and at the mGlu2 receptors, because we know that the primary output of serotonin 2A receptors in the frontal cortex is glutamate release. We also had to look at the overall glutamate receptor binding. In DOB-tolerant animals, we found that, as is expected, that both the serotonin 2A binding capacity but also its capacity to bind G proteins is decreased. We also found the desensitization of mGlu2 receptors, which is interesting because DOB would not directly bind to it. For LSD, unexpectedly, we didn't find any significant changes in serotonin 2 parameters, serotonin 2A parameters, but we also found this mGlu2 desensitization. Interestingly, as opposed to DOB, we found a decrease in overall glutamate receptor binding. So it appears that there are different ways of how the brain achieves tolerance, either directly by adaptation of the 2A receptors or by adaptations of downstream events. Interestingly, in the context of the hypothermia experiments where we applied higher doses of LSD, we did find serotonin 2 desensitization. So again, we have a dose dependency here in terms of tolerance-related phenomenon. Tolerance aside, we were interested in whether there are some therapeutic implications of the repeated application of LSD. We used bulb-actomized rats here. These animals have a disrupted limbic system, and the limbic system is very important for allocating emotional valencies to the environment and memorize the environment accordingly. And if this system is disrupted, animals, for example, are not able to avoid an aversive situation, although there is a way of avoiding it. How is this measured? You see it in this overall setup there. An animal hears a tone, and after this tone, a current is being applied. This current is not painful, it's not harmful, but it's a strong tickle, and animals would rather want to avoid it. Whenever the animal hears the tone, it actually can escape the currents by jumping to a pole. The boot-victimized rats, however, are not able to do that. But if we give them antidepressants subchronically, they become able to learn and to escape the aversiveness. Therefore, we were interested in whether LSD would do the same. And indeed, it did. Interestingly, the healthy animals that received LSD did not benefit from LSD. So this seems to be a specific effect, an ameliorating effect for the pathological condition. We also had a look into the brain, this time into the hippocampus, because it's part of the limbic system and it's very important for learning. We found that bulbectomy is associated with the desensitization of serotonin 2 signaling here, and that LSD would normalize this. Again, in the healthy animals, LSD rather desensitized the receptors, so the effect of LSD is again specific for the pathological condition for the depressed animals. You get this normalizing effect. Serotonin 1A signaling and overall metabotropic serotonin signaling didn't show any regulatory activity here. So it was something specific to the serotonin 2 family. Our research aside, we'd like to discuss with you a basic phenomenon or parameter of pharmacology. It's called affinity, and actually it refers to the concentration of a drug in the synaptic cleft that is needed in order for the drug to bind to a receptor and induce activity. But let me explain how we measure it. If you're interested in the affinity of psychedelic X for the serotonin 2A receptor, you would have these little valves, we call them. There are a lot of cells in there, and we would apply a radioligand to it. And this radioligand would bind all the serotonin 2A receptors. We then apply a little bit of psychedelic X, and this displaces the radioligand from the receptor. We would then increase the dose, the concentration of psychedelic X, until no radioactivity is left. And then we can draw these nice graphs here, and by convention, the affinity of psychedelic X is the concentration in the synaptic cleft, or in this well here, at which half of the radioligand is being displaced. This is the affinity. Why am I telling that? Because it's important for microdosing. It's been suggested that you need about 40% of your serotonin 2A receptors occupied for a microdose effect to occur. The thing is now, psychedelics are not selective for the serotonin 2A receptor. Our psychedelic X, for example, prefers the serotonin 1B receptor before the serotonin 2A. And this means if you microdose yourself and you want 40% of your serotonin 2A receptors occupied, then you will have about 80% of this other receptor occupied. And then you would have to ask yourself, what is it actually this other receptor does? Where is it? In the brain, in other organs of the body? And these are important considerations for safety implications, especially for long-term application. Taking our message, what have we learned today? Tolerance to LSD depends on the interval of application. The smaller the interval, the more likely animals become tolerant. We also learned that tolerance depends on the dose. The higher the dose, the more likely tolerance occurs. And we also learned that tolerance develops differentially. That means some of the effects would become more easy tolerant than others. And this again is important for safety implications and side effects and how they would develop over long term. We also found in terms of mechanism that tolerance is associated with desensitization processes which affect the serotonin 2A, serotonin 2 family, but can also be complemented by adaptations and downstream events and interaction partners such as within the glutamate system. Short-term application of LSD might have beneficial effects when wanting to integrate something, when wanting to integrate aversiveness, when going through things you have experienced and want to learn how to behave next time in a better way. And as to this, of course, this might be relevant for therapeutic approaches with these drugs and depression, but also in other disorders of the psyche. The last slide actually taught us scientists that when we do microdose studies, it's probably a good idea, especially if they are long-term, months perhaps, that we don't just measure psychological output, but also look at other organs which express receptors the given hallucinogen psychedelic has high affinity for, to see whether there are adaptations and whether side effects might be expected as to these physiological processes. That's it from my side. Some thank yous, particularly to the Institute of Pharmacology who made all of this happen, and to my doctor father, Volker Höld. Thanks for all your support, appreciate it. If you are interested in tolerance to LSD, please read our review, it's very comprehensive. And if you're interested in my research in general, please follow me on ResearchGate, and you will be updated as to all research that is coming out soon. Thanks for your attention, and take care.
I guess I should proceed here. Thank you, Tobias, very much for your enlightening lecture. But however, a very important research, and I think we have to look deeper in these issues in the future, especially in that respect, what you mentioned, that everybody is fixed on the 2A receptor, but other receptor might be activated too. And especially if you take a drug on a chronic basis, that might be very important to look at for these other receptors too, which might behave differently if you activate them on a chronic basis. Mm-hmm. And so, yeah, in part, Nilofar's research was in that direction, looking at first for the general effects of very low doses of LSD. And right now they were also looking in the same experiment of on the effects of repeated dosing every fourth day, which is kind of nearing the so-called Fadiman schedule in respect to microdosing. And so there might be very interesting results coming up from that study. But I also want to mention that the background of the study was not explicitly as much microdosing. It was mainly looking for chronic administration of LSD to gain anti-inflammatory effects, which have been shown for LSD in the last 10 years. But let me now introduce Nilofar Family, a researcher, also a PhD, who has done research with LSD before in respect to language patterns during sessions. And afterwards, she became the meanwhile director of Eleusis Benefit Corporation, a study group which also partially consisting of investors looking out for using LSD for clinical purposes, especially in respect to Alzheimer's disease. And so the first step was a phase 1 study to look how much side effects are produced by very low doses of LSD, if any, and can that be administered in a safe way on a chronic basis to healthy older adults. So the average age was kind of 60 or something of the experimental subjects. So now, Nilofar, please start with your lecture. Where I welcome you.
Thanks, Torsten, I think. Hi, I'm Nilofar Family, and before I begin my talk, I would like to thank the ICPR organizing committee for making this event happen despite all the challenges we've all been dealing with one way or another related to COVID-19. And thank you for giving me the opportunity to share some of our work here. I'll be talking about our LSD microdosing safety and tolerability trial, which was published at the end of 2019. The paper is freely available online, and I'll I'll circulate the link in the chat. Before we start, I want to declare my conflict of interest. I am an employee of the sponsor of this work, Elucis. Elucis is a clinical stage life science company based in the UK and in the US. Our mission is to unlock the therapeutic potential of serotonin 2A receptor agonists. The work I'll be presenting today was carried out as part of a clinical trial in 2015. This phase 1 trial was a first clinical step in looking at very low doses or microdoses of LSD for the treatment of Alzheimer's disease. I won't be going through the rationale behind the trial. I'll be walking you through the trial design and some of our results. You'll find details on the connection between LSD and Alzheimer's in our recently published white paper, where we bring together converging lines of research that support LSD as a candidate drug. Please do email me directly, and I'd be happy to send you the white paper. To our knowledge, this is the first clinical trial to investigate microdoses of LSD. The question we addressed was, is regular administration of microdoses of LSD tolerated and safe in an older, healthy adult population? As part of standard phase 1 requirements, we also carried out pharmacokinetic measurements. We assessed changes in cognition and evaluated dose response. We conducted many assessments in this trial, and I've listed them here. Today, I am going to focus on these 4 assessments, namely adverse events, pharmacokinetics, cognition, and subjective effects. The trial was double-blind and placebo-controlled. We recruited 48 participants between the ages of 55 to 75, 32 of whom provided blood samples for PK analysis. Each person was screened within 28 days of baseline, and once they were randomized, they received either placebo, 5 micrograms, 10 micrograms, or 20 micrograms of LSD on 6 occasions. So it was the same dose of LSD on each occasion, separated by 3 drug-free days. This was followed by a 1-month follow-up. Eligibility was based on medical history, physical exam, responses to the CSSRS, suicidality questionnaire, and the SCID-CT, which was modified based on Johnson et al.'s 2008 safety guidelines for hallucinogen research. Key psychiatric exclusion criteria were generally the same as what you see in most studies with psychedelics: an exclusion of lifetime psychosis or first or second degree relative with psychosis. We excluded smokers and did not allow certain concomitant medications, including lithium, SSRIs, SNRIs, MAO inhibitors, or antipsychotics. In total, 27 male and 21 female volunteers with an average age of 63 years and body mass index, or BMI, of 27 enrolled in the study. Age, gender, and BMI were balanced across dose groups. Moving quickly into our methods and data, as part of our safety assessments, we recorded adverse events at each and every visit. Here's a table of all adverse events. I don't expect you to go through all of them. This is published in the paper that came out in 2019. But I'm putting it up here to illustrate the sheer number of adverse events that we had. Now, the placebo group had a very high number of adverse events. It's unclear why. One interpretation would be that the profile of LSD is well known to the public, and there was a sort of expectancy bias. And I can confirm anecdotally that there was a high placebo response. Some participants were reporting effects to me in conversation that were at times quite surprising once the dose groups were unblinded. Clinical review showed that the frequency of adverse events in the LSD groups was similar to placebo, and none were severe in intensity. The only minor clinical difference between placebo and active treatment groups was the number of headaches reported. Transient headaches have been reported in previous studies on classic psychedelics, so this was not a big surprise. Blood samples were collected from 32 participants total, 8 from each dose group prior to dosing and then at 6 time points after dosing. This enabled us to plot plasma levels of LSD over time. And these are the plots for 10 micrograms of LSD on the top at dose 1 and dose 6, and for 20 micrograms LSD on the bottom. These are in picograms per milliliter. Drug levels were under the lower limits of quantification for the 5 microgram group overall, as well as for one of the 8 participants in the 10 microgram group. The Cmax for 10 microgram LSD at dose 1 and dose 6 were 305 and 309 picograms per milliliter respectively, and this had a peak at 30 minutes post-dosing. The Cmax for 20 micrograms was 440 picograms per milliliter and 504 picograms per milliliter, with a peak time at 0.5 and 0.7 hours past dosing for dose 1 and 6 respectively. The ratio between 10 and 20 microgram dosing groups for Cmax and AUC average 1.5 to 1.7, indicating a dose to plasma level positive relationship, but deviating from linearity at these low doses. To test cognition, we used validated tasks from CANTAB, and we used a test for reaction time, visual memory, visual attention, and spatial working memory. We conducted a one-way ANOVA followed by Bonferroni adjustments for multiple comparisons on the CANTAB assessments, and there were no significant differences between treatment groups on any day. To measure subjective effects, we had participants answer a 22-question survey, including questions like, do you feel high? Do you feel good effects? Do you feel bad drug effects? Do you feel dizzy or energized? And they answered these relatively frequently throughout the day. We also used the 5-DASC at the end of each dose day and looked both at the 5 original dimensions as well as the 11 subscales. The maximum values of the subjective effects VAS and the 5-DASC assessments were stable across repeated measures as determined by two-way mixed model ANOVA. Looking, however, at changes in these parameters based on dose, vigilance reduction from the 5-DASC and 3 questions from the subjective drug effects VAS, which were, do you feel bad drug effects? Do you feel dizzy? And does your body feel different or changed? Showed a significant positive linear relationship with LSD dose. One possible interpretation of these results is that participants felt more fatigued by the setting of the study under the influence of LSD due to heightened suggestibility. The participants were where they did these assessments. Interestingly, the increase in vigilance reduction was not observed in the participants in Bershad et al.'s 2019 microdosing study. And also the reduction in vigilance that we saw in our study didn't affect performance on the cognitive tasks. However, vigilance reduction has been reported in previous studies of high-dose LSD. So looking at Lichty, Dolder, and Schmidt in 2017 or Schmidt et al. 2015. So this may be a dose-independent subjective effect of the drug. Our results suggest that the administration of low-dose LSD is safe and well tolerated by this population during the limited 21-day period that we studied. The increase in headaches were mild or moderate suggesting that microdose LSD would not impede day-to-day activities. Evaluation of cognitive and behavioral outcomes indicates a favorable safety profile overall, further supporting the feasibility of periodic LSD administration up to 20 micrograms. And we found a positive increase in a handful of subjective effects relative to dose, and these will be worthy of looking into in future studies of microdose LSD. Overall, our studies appear to justify exploratory patient trials, which are currently under development at Eleusis. And finally, none of this would have been possible without the help and support of all of our staff at the clinical site. So I'd like to say a big thank you to everybody that was involved in this clinical trial. So I'm happy to open the floor to any questions in the chat that I have not addressed up till now. And I thank you for your attention and look forward to the Q&A.
Very good lecture about a large study, as far as I understood it. It took quite a while, and it was heavy work on a clinical level, and trying to get hold of all the participants and all that. And so I'm happy that you have gone through and found something. So let me just tell the auditorium that you were right in mentioning that the vigilance reduction scale was kind of measuring something even with higher doses of LSD, which are not reducing your vigilance. And so therefore it might be that that scale is not really very good. But I don't know what we should do right now. Should we do question and answers here? I heard that we can have one question. So what should we do? I mean, let's see what we have here.
There are a few questions for
Carsten,
yes.
considering that we've been delayed, I would suggest that you all go to the meeting room where you have about 45 minutes in maximum to continue the Q&A. I've posted the link in the chat. Thank you.
Okay, we do that. Bye-bye.
Super. Bye-bye.
Super. Bye-bye. Bye.